Hello, everyone, and welcome to our webinar, Precisionary Instruments. Today, we are thrilled to host Dr. Greta Vargova. Greta Vargova is from University of California, or UC Riverside, but she actually grew up in Slovakia, where she pursued her undergraduate studies in molecular biology, followed by her PhD in neuroscience. And during her PhD research at the Slovak Academy of Sciences, Dr. Vargova co-developed, applied, and characterized several types of adeno-associated viral vectors expressing human truncated tau and red fluorophore mCherry in equal ratios. So these are AAV vectors, are tools developed to induce Alzheimer's disease-like pathology in wild-type mice. So she worked to develop a type of mouse model for studying Alzheimer's. She performed two-photon calcium imaging in awake mice to investigate the effects of developing neurodegeneration on cortical circuits. And she's also had the opportunity to participate in long-term research at Cold Spring Harbor Laboratory, where she studied the role of cortical feedback projections in sensory perception. And currently, Dr. Vargova is a third-year postdoc researcher at UC Riverside and the laboratory of Professor Hongdian Yang, with whom we have had a long relationship with at Precisionary Instruments. There, she studies somatosensory perception and cognitive flexibility, and they are currently developing a novel tactile-based rule shift behavioral assay. So today in this webinar, we are thrilled to host her. She's going to present their most recent work where they show a link between the locus coeruleus activity and behavioral flexibility in mice. And with that, Dr. Vargova, I'm going to hand it over to you.
Thank you so much for the nice introduction. Can everyone hear me? Can you hear me well?
Yeah.
Because I put headphones, okay. Because there is some noise in the background. All right, so welcome everyone, and thank you, Abby, again for the nice introduction. And just like you mentioned today, I would like to talk about the involvement of locus coeruleus in cognitive flexibility, which is our recently published work. The human brain is a complex and dynamic system that allows us to perceive and process. Just one second. Okay. The human brain is a complex and dynamic system that allows us to perceive, process, and adapt to a rapidly changing environment. One critical aspect of cognitive processing is cognitive flexibility, which is the ability to shift attention and to be able to modify one's behavior in response to the changing environment. Cognitive flexibility can be critical in everyday life. And now I would like to give you just one simple example from life where cognitive flexibility could be useful. So imagine you are a driver here in the US and you are used to driving on the right side of the road, while let's say you happen to travel to the UK, well, you will have to drive on the other side of the road. You know how to drive, you know the rules, but still you have to adapt your behavior based on the changing environment. So this was just one example of where cognitive flexibility could be critical in everyday tasks. However, it is the underlying neuronal mechanisms of cognitive flexibility are still not fully understood. In recent years, scientists have paid increasing attention to a small yet very powerful nucleus called, or yet very small and powerful structure in the brainstem that is called the locus coeruleus. It was discovered in the 19th century, and it was described as this blue spot, these two tiny structures in the human brain. And it is even smaller in the mouse brain. It's only, the locus coeruleus is only has about a diameter of 300 microns and contains about 1,500 cells. However, it is the most interesting fact about LC is that it has widespread axonal projections, as you can see on these figures, widespread axonal projections that innervates all cortical regions and all cortical layers. Therefore, it has been shown that it is involved in many neuronal and physiological functions such as motivation, arousal, sleep-awake cycle, stress, flexibility, learning, or memory. And also it has been implicated in normal aging. It is one of the first brain areas that is affected in Alzheimer's disease and Parkinson's disease, and it has been also shown that it is implicated in other neurological disorders such as anxiety, ADHD, or post-traumatic stress disorder. Now, I would like to give you one example of a study that showed how LC could be involved in cognitive flexibility or reversal learning. So this group of scientists trained rats for an auditory reversal learning task, where this behavioral task had two stages, baseline and reversal. In the baseline stage, rats had to respond to sound A in order to receive food delivery, while they had to ignore sound B. Now, once rats learned this task, they reversed the rule, and now rats had to respond to sound B in order to receive food reward and ignore sound A. And in this set of experiments, they optogenetically tagged LC neurons, meaning that they expressed channel rhodopsin in TH positive cells of the LC, and they, during the reversal task from day one, they optogenetically paired the new targets, so sound A, sound B, with an optogenetic stimulation. And in this paper, they show that LC accelerates reversal learning, meaning that here in this figure, the black line indicates the control group, so the performance of the control group in the reversal learning task from day one, while the blue line indicates the performance of the test group. And this figure shows that LC stimulation accelerates reversal learning, meaning the improvement in the behavior or in the reversal was much faster in the test group, where they stimulated LC neurons compared to the control group. However, it is still unclear how LC, or more specifically, the type of LC activity is linked to the degree of flexible behavior. And so we developed a well-controlled behavioral task to probe flexibility, cognitive flexibility in mice. So it is a rule shift, tactile-based task where mice are head fixed and water deprived. We have two rules in the task, either left go or right go. And the big advantage of this task is that mice need to adapt to the new rule within the single session. On each trial, one of the two whiskers is stimulated, either the left or the right whisker. Now, let's say if we start with the left go rule, as it is shown here in block 1 on this figure, that means if it's a left go block, that means that in the go trials, the mouse receives whisker stimulation on the left side and needs to lick in order to receive water reward. While if the mouse receives in a no-go trials a whisker stimulation on the right side, the mouse needs to withhold licking. And in block 2, we changed the rule, meaning that now the mouse needs to lick in the go trials for the right whisker stimulation in order to receive water reward, while in the no-go trials, the mouse receives left whisker stimulation and the mouse needs to withhold licking. In general, we have 2, 3, or 4 blocks, meaning that the third block would be the same rule as block 1. It is random what side do we start with, so it could be for each mouse is trained for the left go, right go, and right go, left go type of session. Based on the trial outcome, we have 4 different trial types. If the mouse correctly licks during the go trial, we call it as a hit, and the mouse receives water reward. If the mouse fails to lick during the no-go trial, fails to lick during the go trial, it is a miss. It is not rewarded or not punished. if the mouse incorrectly leaks during the no-go trial, it's a false alarm and it is punished with a timeout. The mouse correctly withholds leaking during the no-go trials, it's a correct rejection. It is not rewarded or punished. During the session, we have few catch trials, meaning that during the catch trial, we don't have any stimulus presentation and the mouse needs to withhold leaking. Here is our trial structure, which is 6 seconds long. At the beginning of each trial, we have this sound cue, which indicates the trial start. And then during the go and no-go trials, we have a 200 milliseconds whisker stimulation, either on the left or on the right whisker. As I mentioned before, during the catch trials, we don't have any stimulation. And in the go trials, the mouse has 1 second to respond to the whisker stimulation in order to receive water reward. The training process takes a couple of weeks. For any further analysis, we excluded block 1 since our question is focused on cognitive flexibility or reversal learning, while block 1 does not involve any rule change. So this plot down here describes the switch performance of 11 mice in block 2. Like I said, training process takes a couple of weeks. We consider mice to be trained once they reach 65% threshold, and the switch performance has to be about 65% for at least 2 consecutive days. The learning process was associated with increase in the correct rejection rate, not in the hit rate. Trained mice. We noticed that even trained mice show varying levels of flexibility or varying levels of switching behavior in this task. So here are 2 example sessions from the same mouse. Each of these sessions contain about 400 trials and 3 blocks. So this gray bar or line indicates the rule switch. I forgot to mention at the first figure that this gray bar, or where we start or we change the rule, we have 5 to 10 queuing trials at the beginning of each block to indicate the mouse that the rule has switched. So in these 2 figures, the black line indicates the fraction of correct trials. The orange line is the correct rejection rate, and the magenta is the hit rate. And these arrows up here indicate the switch point or the switch trial. So if the fraction correct passes the threshold of 85%, we call it as a switch point or switch trial. Now, these two sessions are different in a way with the degree of flexibility, meaning that in this first particular session, the mouse was able to switch at the beginning of block 2, while in block 3, the mouse had more time to be able to adapt the behavior to the new rule, while on the next day, the mouse was only able to switch at the end of block 2, but immediately switch in block 3. Now, if we look at the switch performance from all groups or from all mice and from all blocks, then we can see that there is a strong negative correlation between switch point and switch performance. And based on this plot, we notice that there is separation around trial 50. So we determined or we defined blocks where the mouse switched before trial 50 as a fast switching block. And we defined blocks where the mouse switched after trial 50 within the particular block as a slow switch. And as expected, if we compare the switch performance of the fast and slow switching blocks, then we can see that the switch performance is significantly higher in fast switching blocks compared to the slow switching blocks. Like I mentioned in the first slide, each mouse is trained for both types of sessions, which means that it can be left-right or right-left. So we asked whether there is any difference in these two types of sessions, any difference in the switch performance, number of clicks, or reaction time, or if there is any side bias in these mice, these 11 mice that I just described to you, and we did not find any significant difference in the switch performance, number of clicks, reaction time between left and right switching blocks, not in the fast switching blocks or in the slow switching blocks. Now, since we noticed that there is significant difference in the switch performance of fast and slow switching blocks, we also asked whether there is any change in the motivational state of these mice. And so we looked at the number of clicks and reaction time and compared between fast and slow switching blocks. And these results show that there is no significant difference between fast and slow switching blocks in the regarding clicks or reaction time. These data that I just show you demonstrate that mice show varying levels of flexible behavior in this novel tactile-based rule shift task, and it's not dependent on the motivational state of the mouse. So next, our goal was to record from LC neurons from these mice that were well trained for this tactile-based rule shift task. So for this type of experiment, we crossed DBH3 mice with AI32 mice. So the DBH mice express Cre recombinase in noradrenergic neurons, in LC neurons, and then AI32 mice express channel rhodopsin upon the exposure to the Cre recombinase. As a result, we have mice where channel rhodopsin is expressed in TH positive cells in LC. So for this experiment, or for this experiment and other type of experiments that we do in the lab, either using the transgenic mice or using stereotoxic injections, we use the precisionary compressome to prepare these slices. We usually prepare between 50 to 100 micron slices. And I think the biggest advantage of this compressome is that it's very fast. Sometimes we have to slice multiple brains, and we have to slice all the brain. So like I mentioned, we use these DBH3 AI32 mice that express channel rhodopsin in TH positive cells on LC neurons, and these mice were implanted with a custom-made tetrode, so we can record spiking activity in the LC. And during this experiment, while mice were performing this tactile-based rule shift task, we also recorded the pupil diameter. Here is one example LC neuron that responds to the optogenetic stimulation. And down here you can see a raster plot of LC spiking activity aligned to the optogenetic stimulation. Each row represents one trial here. And so based on our behavior results, where we showed that mice show varying levels of flexibility, we asked whether there is any difference in LC activity during these two types of blocks. So we compared LC activity during a fast switching block and a slow switching block of the same session. In this raster plot, each row represents one trial. The black trials up here and down here indicate trials of of the previous block, so before the rule switch, while the blue and the red trials indicate trials after the rule switch, so of the current block. And we focused our analysis based on previous results. We focused on our analysis on this 1 second baseline window before the stimulus onset. Now we compared 7 fast and 7 slow switching blocks. So these were paired blocks, meaning that they were in the same session. So if you look at the average activity of these fast and slow switching blocks, before or after, meaning before is 20 trials before the rule switch, and after is 20 trials after the rule switch, then we can see that there is we notice that there is an elevated LC baseline activity during the fast switching blocks, but not in the slow switching block. And so we also directly compared LC baseline activity before and after the rule switch between fast and slow switching blocks. And this figure also suggests that LC baseline activity is increased in fast switching blocks compared to the slow switching blocks. And this figure down here is the quantification of what I just described to you before. So again, we compared 7 paired blocks, fast and slow. So first, if we look at the fast switching blocks, then we can see that there is a significant increase in LC baseline activity following the rule change, while there is no significant difference before and after in the slow switching block. And if we directly compare the 2 types of blocks, then these results show that there is a significant increase in LC baseline activity in the fast switching block compared to the slow switching block. Importantly, these 2 figures on the right show that we can get similar results even if we don't define these blocks as fast and slow switch blocks. So here we pulled all blocks together, and these 2 plots show that there is a strong negative correlation between switch point and LC firing rate. So the first plot is the quantification of 1 second pre-stimulus activity, while the second plot is a quantification of a 0.5 second pre-stimulus activity. So these results show that baseline LC firing rate is correlated with the degree of flexible switching, meaning that fast switching is correlated, or fast switching means higher LC baseline activity. Next, to assess what is the role of LC in cognitive flexibility, we optogenetically activated LC neurons. So again, we used mice that I described to you earlier, these DBH3 AI32 mice, where as a result in these mice, channelrhodopsin is expressed in LC neurons. And in this set of experiments, mice were implanted with an optic fiber in one hemisphere over the LC. We had four test mice and 3 controls. So in the control mice, optic fiber was implanted off target, meaning that LC neurons did not receive any light or very minimal. In this optogenetic experiment, we optogenetically activated LC neurons during go trials, and we optogenetically activated these neurons before the stimulus onset, since in the previous results showed that LC baseline activity is associated with the degree of flexibility, and we optogenetically activated LC neurons in the first 50 trials of each block. To confirm the placement of the optic fiber, we also recorded pupil diameter in this experiment. And as expected in test mice where the optical fiber was implanted over the LC, so these LC neurons received light, the pupil diameter significantly increased compared to the control group. So for this type of experiment, we used mice that were not able to learn this tactile-based rule shift task, meaning that both for the test and the control group, the switching performance was around consistently around 50%. And then this the sign bar indicates where we started optogenetic stimulation. And this figure, example figure, shows that in the test group, after starting the optogenetic stimulation, the switch performance increased. Now, if we compare individual mice, then we have similar results, what I just described to you before. So again, we had 4 test mice and 3 controls. So in the test, in each of the test mice, the switch performance significantly increased with optogenetic stimulation, while in the control group, there is no significant difference in the switch performance before or after optogenetic stimulation. And to summarize what I just described to you, to pulling all sessions or all mice together, these figures or these results show that the switch performance significantly increased in test mice with optogenetic stimulation, while there is no difference in the control group. And as expected, the switch point significantly decreased in the test mice, but not in the control group. And so if you remember from the very first figure when I described the behavior task and how the training process looks like in these mice, then I mentioned that the correct rejection rate is associated with, or like the training or the learning process is associated with increase in the correct rejection rate, but not in the hit rate. So we also wanted to see whether this is true for the optogenetic experiments, and these results show that again there is no significant difference in hit rate before or after optogenetic stimulation in the test or in the control mice, while the correct rejection rate significantly increased in test mice but not in the control group. So this is aligned, like these results align with the learning process of mice in this task. So these results show that LC activation improves reversal learning. And so to conclude what I just showed you before, I showed you that we developed this well-controlled novel bilateral single whisker discrimination go-no-go task that allows well-controlled investigation of varying degrees of flexible task switching. I also showed you that baseline LC activity is correlated with the degree of flexible task switching, meaning that higher activity was associated with faster behavioral switching. And lastly, with the optogenetic experiments, I showed you that activation of LC increases switch performance, suggesting a causal role for LC in behavioral flexibility. I'm happy to take any questions, but if you are more interested, these results were published last year in 2022. And I have one more slide just to thank the whole Yang lab for any support, and I would like to especially thank our group leader Hongdian Yang for supporting this whole study. And I would like to mention that Jim McBurney-Lin, our grad student who collected this whole data set and developed the task, and Machindra, who helped with some histology images, and Edward Zaga, who is also a group leader here at UC Riverside, who also supported this study. And thank you so much for the attention.
Oh my gosh, thank you so much, Greta. So I want to open the floor up. I didn't get any questions in the chat, but if you would like to unmute yourself and ask a question, please feel free to do so, or you can type your question into the chat box. Does anybody have any comments or have a question for Dr. Vargova? Do we have any questions? If not, please feel free to reach back out to us at Precisionary. Sometimes questions come along afterwards, and Greta, we will send them your way if they do. In the meantime, thank you so much. We want to say, it looks like Merrick Schwint, you unmuted yourself. Do you want to comment?
Oh yeah, I had a quick question.
Please.
So it's great work. Do you know where these neurons project in terms of, we know where they project from LC, but with significance to this behavior, do you know, is this where does this neural activity leads to? It's probably some, we know that like orbital frontal cortex is controlling reversal learning. Is this something to do? Is this part of your circuitry investigating?
So specifically in these experiments, what I showed you before, like the type of activity that we see, I want to go to the very first slide, sorry. So the type of activity they see, we don't know like specifically these neurons where to project. We know that LC project, like you said, to the orbitofrontal cortex. It's also possible that like during this task, it modulates sensory perception, since it also projects to the sensory cortex. But I think in the past few years, like people have been, or scientists have been paying attention to the heterogeneity of LC neurons. So I don't know specifically like the neurons that we recorded from where do they project. But I think, yes, one possibility is orbitofrontal cortex and, like I said, sensory perception.
Thank you.
Wonderful. Dr. Quillfeld, I'm sorry if I'm mispronouncing your name, also has a question. He typed it in the comment box, and if it's okay with you, I'm gonna go ahead and ask it for you. He wants to tell you that it was a beautiful presentation and congratulations on your publication. And the question is, isn't the fact that the fast switch performance is better than the slower rather surprising? I can only think that memory can be caused if this difference, working memory, that would fade for the slow group, and that this might involve the MPFC. And what are your thoughts?
That's a great question. So, because sorry, I have to read the question.
Yeah, it's in the chat if you wanted to pull it up.
Yes, I have it. Sorry, just the fact. I think it's an interesting question, and I'm not sure I can answer to this question, but I think it's a bit similar to the previous one. So I think it depends where these neurons project to and like what other areas are involved. Like I mentioned before, it can be also OFC and how LC modulates the somatosensory cortex. It can also be MPFC. Like how is memory involved in this? I'm not sure I can answer to this. I think it's very complex. So I think probably this study, or not probably, but for sure this LC needs to be or like we can study how these different types of neurons in LC would react or would behave in this type of experiment. I don't know.
Maybe it would be the next step of your work.
Yes.
Very nice. All right. In that case, if we don't have any other questions, oh, we do. Dr. Flores has one. And Dr. Flores, do you want to unmute yourself and ask the question? I'd be happy to do that.
Oh, good morning. I had a couple, thank you for this beautiful talk. I'm also working with Dr. Zega using the go, no go task, but using visual instead of whisker. So a few kind of like, sorry, basic questions. The first is like, I was curious if you might see, like, because I'm using a visual task, would you predict there would be similar learning rates and improved searching across different cues between whisker and visual if we also use a similar like locus coeruleus stimulation? That's like my first question. Does the modality matter?
So it's a simple go, no go task, right? So it's not a reversal. Is that right?
Oh, I'm using a visual. So like we're thinking about doing a switch as well. I was just wondering, you said it took a couple of, it took time to actually teach the animals the switch between learning, between the blocks using a whisker stimulation task. I was wondering if you would expect like similar learning across modalities and similar modulation.
So I only have experience with this task, but I don't think that the modality matters. So we never tried using a different, like either auditory or visual stimulation instead of the whisker stimulation. But I think reversal task in general is difficult for mice. So probably you do the same or you will be doing the same that first we train just for a simple go, no go task. Which mice can learn in a week or two, and then the reversal process is the one that takes longer. So like I mentioned, I don't have an experience with other modalities, but I think the training process is going to be similar for you.
Okay. And I guess like my slightly more interesting question then would be, you mentioned that when you're doing the locus stimulus stimulation, you were doing it during the first 50 trials per block, and you did this for a couple of days. First question would be, what's the best way to put it? How long did you see this improvement? Did you only see this in, like, I saw that, okay, there's an initial improvement, and you kept seeing this improvement as you had the simulation, but it looked like it, at some point, yes, thank you for going back to data.
Yes.
If you could go to, I think, the next image, the next figure. Yes, this one. Oh, sorry. I'm sorry. I guess it's maybe, sorry, previous.
Okay.
Thank you so much.
Oh, sorry.
Yes, the days. You see that with simulation for days 1 through 5, with the increasing simulation, you do see like a bit of improvement, but it almost looks like it kind of plateaus, like it improves, but it kind of plateaus. I was curious about what your thoughts were in terms of, like, I guess there's only so much you can improve. Like, instead of like the animals kind of plateau to 70%, instead of going from like 70%, 80%, 90%. Any thoughts on like why there might be a plateau with like all of these like stimulations, 50 first trials per block across these multiple days?
So, yeah, it's an interesting question. So like you mentioned, there is a immediate increase in the performance. So I don't know if I mentioned when I was talking about this figure. So we compared the last 5 sessions from the baseline period and then 5 sessions after the baseline period. So we didn't check like what happens later. So I don't know if it can still increase, but my guess is that what you were saying, that it plateaus at some point. I think it can be also because I'm not sure, or like whether it would help to, we are only stimulating a subset of neurons. Like what would happen if we would be able, I don't even know if it's possible, if we would be able to activate all the neurons, whether it would help. But I think there are other aspects of the behavior. So it's not only LC is definitely involved in this task and is needed for the cognitive or the rule switching or the reversal switching, but there are other brain areas that are involved in this. So it's not purely based on LC.
Mhm.
So I don't think that activating LC would, I don't know, increase the task, let's say to 100% or the performance to 100% or something like that. I don't even know if it would be possible.
No, thank you so much for your patience, and I will definitely follow up with you with additional questions as I read your paper. I appreciate you.
Thank you so much for the question.
Wonderful. I think that's a great note on to in your webinar. Thank you so much, everybody, for coming, and I hope you all enjoyed it. And thanks again to Dr. Greta Vargova.
Thank you so much for having me.